This is Dr. Malati from Department of Life Sciences. This session we will be dealing about different types of RNA. In general, RNA, which is a polymer of nucleotide which is made up of ribosugar, phosphate and bases such as adenine, guanine, cytosine and uracil. It's a polymeric molecule which is essential for different biological roles, which includes coding, decoding, regulation and gene expression, and which is very stable molecule when compared to DNA, and this is very useful for synthesis and expression of new genes and all, which includes expression of different proteins. So we have different types of RNA, which includes coding RNA and non-coding RNA. The coding RNA, which includes mRNA alone, this mRNA will be expressed as different proteins. So these proteins only will carry out different biological process. So all the genes which are expressed will be expressed through mRNA molecule, and these mRNA molecules will be forming a new protein, and those proteins will have biological function. Under non-coding RNA, we have ribosomal RNA, transfer RNA, small nuclear RNA, small nucleolar RNAs, PV interacting RNAs, microRNAs, long non-coding RNAs. Now we'll start with coding RNA, which includes mRNA molecule. The mRNA molecule which carries the genetic code as such from the DNA, which will be exactly complementary to that of the gene, which is going to be expressed as protein. Through the mRNA molecule, we express new proteins. The proteins will have different biological roles. The coding sequence of mRNA will determine different amino acid sequence in a particular protein produced. So each codon will pick up the amino acid through the tRNA molecule, and that amino acid will be incorporated as the new protein is synthesized. So once transcribed from DNA, eukaryotic mRNA will be called as pre-mRNA. So before even it gets matured and processed, we call it as pre-mRNA. After it completely processed, it will be called as mature mRNA. So through mRNA only different protein molecules are expressed. The process of converting pre-mRNA to mature mRNA, we call it as RNA splicing. This removes the interons, which is non-coding sequence, and joins the expressing regions or expressing sequence to form mature mRNA. As part of post-translational processing in eukaryotes, the 5' end of your mRNA is having cap, which you call it as guanosine triphosphate nucleotide. This mRNA capping also 3' end of mRNA has a poly A tail. These two regions will serve as the capping and tailing of the mRNA. Both will increase the stability of the mRNA, so that the degradation of our mRNA will be delayed and the enzymatic degradation of mRNA will be restricted. Both the 5' and 3' capping and tailing will increase the time span of the mRNA which can spend in the cytoplasm, so that the expression of mRNA into protein will be more. Under non-coding RNA, we have ribosomal RNA, which we call it as rRNA. The ribosomal RNA has the catalytic component, and this catalytic component only will be used for the synthesis of new protein. In the cytoplasm, rRNA and along with the protein together it forms nucleoprotein complex, which we call it as ribosome, and that ribosome which will go and bind with mRNA and synthesize new protein through translation process. So under eukaryotic cell, the eukaryotic ribosome, based on the mass, it will be divided into two Swedberg units. One is small s, means smaller subunit. The other one is a larger subunit. The smaller subunit is having 40 Swedberg units, and the larger subunit is having 60 Swedberg units. The 40 Swedberg units contains 18S rRNA and also 33 different types of proteins, whereas the 60S RNA, ribosomal RNA, which has 28S rRNA, 5.8S rRNA and 5S rRNA, plus 49 different proteins which are available. So including this 33 different proteins and 49 different proteins, all will be used for expressing the mRNA into protein. Whereas in case of prokaryotic cell, the prokaryotic cell's ribosomes mass is 70 Swedberg units, out of which 30S forms the smaller subunit and 50S forms the larger subunit. Under 30S subunit, we have 16S ribosomal RNA plus 21 different types of protein. And in case of 50S Swedberg unit, 23S rRNA is there and also 5S rRNA is there, plus 31 different types of protein. So 21 different types of protein plus 31 different types of protein, which includes the structure of the ribosome, which will be used for the transcription and translation into protein structures. So the next is very important RNA, which is transfer RNA. This transfer RNA, which includes 80 nucleotide bases. So this 80 nucleotide bases will form a secondary structure, which we call it as cloverleaf structure, which is listed in the diagram. So this cloverleaf structure will contribute for the structure of tRNA. So this tRNA will recognize the codon which is present in the mRNA, and according to the codon, the amino acid will be picked up by the tRNA. This specific amino acid will be incorporated in the newly synthesized protein, and the sequence of codon which is present in the mRNA, that will be read with the help of the tRNA. Accordingly, the amino acids will be picked and the sequence of amino acids will be given to newly synthesized protein. And here, if we see the structure, the 3' end of the tRNA, which will have CCA sequence. So this CCA sequence will act as acceptor arm, which will pick the specific amino acid according to the codon which is present in the mRNA. As we move from the 5' end, the first stem and loop structure which is formed, which we call it as D loop. So this will have dihydroxyl residues. So the modified unusual bases will be there along with methylated guanine. So this particular loop forms D loop, and then followed by that we have anticodon loop. This anticodon loop also is a form of stem and loop structure. This anticodon loop will be interacting with the mRNA codon. So according to the codon which is present in the mRNA, the particular anticodon will be read, and according to that particular anticodon, the acceptor arm will receive the specific amino acid. In the anticodon loop, we will have some unusual bases, which include pseudouridine, dihydroxyuridine, inosine. So these are all the unusual base pairs which will be present in the anticodon loop. After reaching the anticodon loop, little more if we go beyond the sequence, we will have small variable loop. It depends on which kind of tRNA or which different types of tRNA you have. The variable loop sequence will be different. Either it will be 7 or 8 or 9 nucleotide in it. And followed by that, we have another stem loop structure, which we call it as pseudouracil, plus you will have T and CG loop. So this loop will have unusual base pair as such. Followed by that, we will end up with 3' end. The 3' end will have a specialized sequence, which is CCA. So this CCA only will accept the specific amino acid. So whatever the codon which is present in mRNA, which will be detected by means of anticodon, which is present in the anticodon loop. So according to that anticodon, the specific amino acid will be picked in case of the mRNA. So mRNA will have amino acid arm, which you call it as acceptor arm, followed by the D loop arm or D arm, followed by that anticodon arm, then extra arm or variable arm will be there, then T arm will be there. So T arm will have pseudouridine, dihydrouridine, inosine, ribothiamidine. Such kind of unusual bases will be present more in case of T arm. Followed by that, we have other different RNAs which are also equally important for the biological function, which includes small nuclear RNA, which is around 150 nucleotide in size. The small nuclear RNA will be always associated with different kinds of proteins, and it complexes into small nuclear ribonucleoprotein. The major function of this is to process the mRNA. So from pre-mRNA to get converted into mature RNA, we need these kinds of small nuclear RNA. For the splicing process, the small nuclear RNA will be used. And the next is small nucleolar RNA. The small nucleolar RNA is around 600 to 300 nucleotide in size, and these are all the components of nucleolar ribonucleoproteins. So this is very responsible for sequence specific nucleotide modification. For the modification of sequences which is present in different RNAs, this particular RNA will be used. Then we have pi-way interacting RNA whose nucleotide size will be around 24 to 30, and this will be involved in maintaining the stability of the RNAs in the germ layers, especially the genomes in the germ layers, and this also will play an important role in gametogenesis. Then we have microRNAs. MicroRNA size is around 21 to 22 nucleotide, and this 22 nucleotide containing microRNA, which is very important for post-translational gene silencing process. So along with this will be interfering with miRNA and enzyme. So together with miRNA and enzyme will go and block the mRNA being translated. So automatically the mRNA cannot be expressed into protein, and also this has the capacity to accelerate the degradation of the particular RNA. Automatically new proteins cannot be translated, the protein synthesis itself will be blocked. The last, long non-coding RNAs. These long non-coding RNAs are heterogeneous group of non-coding transcripts whose nucleotide length will be more than 200, and the larger portion of the non-coding RNA transcriptome you can see in the mammalian cell. Around 8,000 long non-coding RNAs are present in the human genome itself, and this has many physiological processes. So these physiological processes can be regulated using long non-coding RNAs. Thank you.